There we go. So disclosures include my work with the WFH and the NHF, of course, but I also have worked in drug development over my whole career and have been involved in the development of 5 or 6 novel hemophilia clotting factor products. And so that also serves to inform. And I spent a lot of my professional time working in the area of gene therapy. Okay, sorry, I'm getting some delays on moving the slides, but I think we've got it. Okay, so I think you're all aware how rapid the field has progressed over the past 55, 60 years, where we've gone from really no treatment at all to state-of-the-art therapies that are based upon recombinant DNA technology that really have ushered in the biotechnology field over the past 30, 40 years. And so that began with moving from plasma-derived clotting factors, which, as we all know, were contaminated by HIV, hepatitis C, hepatitis B, and did result in the death and destruction of many members of our community, to recombinant clotting factors, which were much safer, eliminating the risk of bloodborne transmission, and more recently to extended half-life clotting factors. And then we've moved into some new novel therapy platforms, including bispecific monoclonal antibodies, agents which can change the balance in coagulation that are still in clinical testing, and gene therapies still in clinical testing. What's most important, though, is what's on the right-hand side of the slide, because it shows how the treatment has actually changed. The care of the patient has changed based upon the advances in treatment. And so what we can see here is we've gone from on-demand therapies to treat bleeding episodes after they've started, which has resulted in significant joint destruction and sometimes death, to some prophylaxis, more prophylaxis, and now we're entering an era of really 24/7 coverage. And so that's quite a change from what we've had before. So let's start with talking about gene therapy. To do that, I want to talk about what exactly a gene is. And so you're all aware that we have lots of cells in our body. They all do different things. And within the nucleus of these cells are chromosomes. We have 23 sets of chromosomes. And inside of those chromosomes is DNA. That DNA is arranged in very specific ways and sequences, and it results in the formation of about 25,000 genes in each human being. So factor VIII, factor IX, von Willebrand factor, all of those are individual genes. And they provide the instructions to the cell for making the protein. And so with gene therapy, the concept has been around since 1970 or so. The idea is to fix a broken gene, so to speak, a gene that doesn't work, with a gene that does work. And therefore the disease becomes cured. And so gene therapy has always been thought of as a way to cure diseases because you're changing the DNA inside of a person's cells. And for that, invokes the idea that we should cure a patient by fixing the gene. So the question is, how do we do this? And the diagram shown here is a simplified one. It's taking the DNA for the gene we're interested in inserting into a person, packaging it. And the best thing to package it in are viruses. Viruses have been infecting mammalian cells since there were mammals, vertebrate cells for 500 million years since there were vertebrates. And as a result, they're very good at it. And so with gene therapy, we inject intravenously the virus that contains our gene of interest. We've taken the viral genes out. And then that circulates and the virus can home in to the specific cells that we're interested in, which in our case is the liver. We don't understand a lot of what goes on when a cell hits the hepatocyte, the liver cell outer surface. gets in, and then at a certain point, protein comes out, factor VIII, factor IX. So while much of this is not understood, we do understand that we can measure protein, and the protein can have therapeutic value. So the history of gene therapy is young, and we have progressed through a first wave of clinical trials that I had been involved in back in the end of the last century, beginning of this century. They didn't work, but they did inform on what some of the hurdles were and obstacles in achieving a reliable gene therapy. So we went back to the bench for a number of years and began in the 2010s to see some proof of concept, initially in hemophilia B. And now we have a whole current wave of trials, some of which I will discuss, that are showing a variety of different results. So this is another diagram, and it shows what happens when gene therapy is administered. And so the viral vectors that are carrying our gene of interest are just infused, just as anything else would be infused into a vein in a person. And in our case, many of those virus particles will home to the liver, where they'll set up residence. And they'll set that residence up by getting into these cells in the liver and gradually making their way to the nucleus, where they release their DNA and form something called a stable episome, which is a circular piece of DNA. This contains the factor VIII or the factor IX gene, and it allows messenger RNA, mRNA, to be expressed. You may remember that from high school biology. And the mRNA then is able to make protein. And that protein, factor VIII or factor IX, is secreted. So it's a remarkable situation, taking advantage of the body's own machinery to make a new gene, a transgene. What's the goal? Well, the goal really is a functional cure, as I mentioned. And so some of my colleagues, including Mark Skinner, who was the first author here, put together a paper a year or two ago, a couple of years now, that describe how to get to that goal. And they look at it in two ways. One is in terms of health equity, getting access to treatment, and what does that do for a person? And the other way is in terms of a functional cure, looking at the clinical outcomes. So as we start at the bottom here, which affects the majority of patients with hemophilia in the world, the goal is to prevent premature death, and on the clinical side, to induce survival. And then with some factor concentrates available, to decrease the amount of joint damage that occurs, improving quality of life along the way, and allowing for an increase in quality of life, participation in work, family, career, and a more unrestricted lifestyle. And so as we move forward on the treatment side, it really involves ultimately achieving normal hemostasis. And that's really what a functional cure is, normal hemostasis. So this principle has been applied to hundreds of different diseases. This is an eye chart that I recognize. I'm not expecting you to actually read anything on here, but I want to point out the number of different gene therapy clinical trials, largely with AAV, that have been undertaken over the last 10 plus years. And what we've got in the outer ring is preclinical work. And then in the next ring, the light blue ring, is early stage phase 1-2 clinical trials, pivotal clinical trials, gene therapies that have been filed, and then gene therapies that have been marketed. So what you can see is that 2 gene therapies have been marketed to date. One is Luxturna for a disease that causes blindness in the eyes, and it's injected locally into the eye. And the other is Zolgensma for spinal muscular atrophy, which is a muscle wasting disease that affects infants, usually death within the first year and a half of life. And this can be administered systemically and truly does prevent progression of the disease. So a remarkable drug in and of itself. Hemophilia is up here, these two columns up here, and you can see there's a lot of activity. One has been filed, BioMarin's hemophilia A, and the other one, Unicure, right here, is about to be filed for hemophilia B. And so we are in the midst of a real renaissance in the development of gene therapies, but there still is a lot of work to do. If I look at the whole field, hundreds of studies have already failed preclinically, clinically. Some have even failed after licensure. So this is a tough area to work in. We don't have much of a therapeutic index, meaning the side effects of the gene therapy come with any efficacy, come right along with any efficacy. We see a lot of variability, low values, high values for the gene of interest. And in at least the case of factor VIII, a lack of durability and inconsistent reliability. Some patients don't respond for unknown reasons. And then there's always the question of unknown toxicities. So how do we think about the gene therapy benefit risk ratio? That's something that a number of groups are giving a lot of thought to. It really is the efficacy balanced against safety. And so we're looking at gene therapy as a once and done, a cure for the disease, versus whether it isn't a once and done, but has a durability of 5 to 10 years, or it doesn't even produce a good result. So that's all factored into the benefit risk ratio. One also has to look at what the alternatives are. In North America, Western Europe, we've got state-of-the-art therapies that allow patients to live relatively normal lives with minimal impact from their hemophilia. The rest of the world, not so much so, but that's part of looking at the benefit risk in different parts of the world. And then we've got a lot of knowns and unknowns, and this is a useful framework to look at benefit risk. What we know, we know. There's a lot that we know about gene therapy and the benefits and the risks, and there's a lot that we know we don't know about it, but it's being investigated. There is also, though, the unknown unknowns. What we don't know, we don't know. And those are the surprises that appear whenever we're developing a new technology and attempting to determine what its benefit risk ratio is in humans. Oops. And so toward that end, the FDA held a meeting last September that really looked at toxicity associated with AAV therapy. And in that meeting, they went through a number of different clinical situations. They looked at hepatotoxicity, which we see in hemophilia, other diseases where higher doses of drug are given, the gene therapy are administered, have increased risks of toxicity, including liver failure and death. And then there are other various diseases that also appear associated with high doses of gene therapy. And so this was really the first systemic look at what the safety profile is for gene therapy in a variety of different diseases. Why are we seeing this safety profile? We're seeing it because as a field, we are continuing to move up in dose in order to achieve efficacy. And so we're now at doses that are in the 10 to the 14th range. That's 100 trillion vector genomes per kilogram. And so if you are a 100 kilogram person, that is 10 quadrillion vector genomes going into your body. Remember, I told you that there were about 30 trillion cells in the body. So there's a lot more viral vector going in than we actually have cells in the body by many orders of magnitude. And that causes toxicity with such a large bolus of these viral particles. So let me turn now to specific gene therapy studies. I'm going to first talk about the largest gene therapy study that actually has ever been done, including in hemophilia, but in all different diseases, and that's the studies with garaktakojin, roxaparvovec. Three New England Journal papers came out. The first one on a phase 1/2 initial results at 1 year, second one on a multi-year follow-up, and then the third one on the phase 3 clinical study with the 132 patients. And so I think this might be a little blurry, but what I want to show you here is a couple of things. The orange bars are all of the patients in the phase 3 study, the phase 1/2 study over 5 years. And so what you can see is an early peak of activity. This is the factor VIII activity on the y-axis, and then the duration for 5 years on the x-axis. Peak of activity in the first year, and then gradual lowering of the activity for this high dose. And you can see that in the table here. Year 1, it's at 60% on average, which was a great average. 60% factor VIII levels would be in the normal range on average. But by the end of year 2, it's dropped in half. By the end of year 4, it's dropped in half again and continues to drop by the end of year 5. And so that drop indicates that we lack some durability in this study. This was a small number of patients, so it was important to study this in a phase 3 trial, and that's exactly what was done. It's called the GENERATE study, and it is administering the same dose that I just described of garaktakojin, roxaparvovec, to adult men with severe hemophilia A. And so they then analyzed the results after 1 year, after 2 years, and I'll show you a little data from 3 years. First of all, it's really important to point out that this, along with the other gene therapies, does indicate that it works. We're able to get a gene into the cells and secrete enough factor VIII to make a difference in the annualized bleeding rate. This is the before picture. This is the after picture. The annualized infusion rate, 130 infusions before per year, down to nearly 0 infusions afterward with the median. And the number of treated bleeding episodes as well is also greatly reduced. So there's no question, therapeutic transgene is getting into the cells, protein is being made, being secreted, and functional factor VIII is working to prevent bleeding episodes from occurring. So that's really important to point out. Now, what about the activity levels? Well, this is in the phase 3 study looking at the activity over the course of a year. And you can see there's always an early peak, but then things settle out. And so by the end of the year, we are at a median of somewhere around 20% and a mean that's somewhat higher. And the mean is reflected in the fact that you've got some very high values, over 200% factor VIII for a period of time here. But the medians are the more important ones to look at. So around 20% and change. This is looking as a function of time, the factor VIII level on the y-axis again. And so it's not only looking at the first year results, but it's going out to look at all of the patients over 2 years. And so you can see a gradual drop off over 2 years. And then there's a subset of 17 patients where you are continuing to see a drop off over 3 years. And so the question of durability that was found in the phase 1/2 study remains in the phase 3 study. And it does look that factor VIII is dropping off. You can also see very high variability based upon these bars that indicate the interquartile ranges. So some of the range of factor VIII activity ranging from 0 on up into the 200% range. And then reliability, where a proportion of patients did not get any benefit from the gene therapy. So this trial has demonstrated a considerable amount of data. Now, what about the safety side? Well, these liver enzyme elevations that I've mentioned before do remain, and these enzyme elevations were present in 89% of the 132 patients or so. There were a few other adverse events, but the most important one here is the elevation in the liver enzyme because it indicates some damage to the liver. There also were some relatively high liver enzyme elevations. So about 8% of the patients had elevations at least 5 to 20-fold above the upper limits of normal. No evidence of any thrombotic events, clotting, excess clotting, no evidence of any neutralizing antibodies or inhibitors to factor VIII as well, and no evidence of any malignancies during this trial. So the conclusion of the authors from this phase 3 paper are very straightforward. It provides endogenous factor VIII production, and it significantly reduces bleeding and factor VIII concentrate use relative to prior factor VIII prophylaxis. But the trial also raises unanswered questions, which I've listed here. What is the role of the steroids and the other immunosuppressives that were being used in the trial to try to dampen the ALT, the transaminase liver enzyme response? And why was it required for a median of 8 months in the majority of participants? What is the cause of the elevated ALTs? That's not at all clear. And it appears there are probably multiple causes and not a single cause. And really the cause can only be answered by getting biopsies of the liver after treatment, but very few of those have been done. The cause for the loss of activity, not at all clear either, but that would come from doing liver biopsies and understanding what's happening inside of the cells from the patients who received the therapy. And then 14% really got a minimal kind of response or no response. And what happened there? There's no known reason for that. And so that's an important question to answer. So the phase 3 trials given us a lot of information, has also raised a lot of questions. This will be the basis for the possible approval of this drug in the coming year. So what are other groups doing? Well, here's data from a couple of years ago from Bayer Healthcare. They did a small trial with 3 different doses, 2 patients per dose. And you can see some evidence of a dose response curve, the low dose down here, the intermediate dose co-mingled with the low dose, and then the high dose showing an increase for both. But you can see that there's a large gap between the 2 patients who had received the same amount of vector. So variability shown here. We can't say much more about this study because the patient numbers are so few, and we can't say anything about durability at this point because the patients haven't gone out far enough. We do know that some of the patients required steroids, and so there were some transaminase elevations there. Here's a study that has 18 patients total in it, and this is a difficult graph to read. But what it shows is the administration of an AAV factor VIII in a study called SPK 8011. And it shows these high peaks early on. This is looking at the factor VIII activity as a function of years. And so within the first year, we've got a lot of noise, a lot of settling down. But then we also see one patient lost all of their activity. This patient also was on the road to losing all of his activity. And the rest of these patients look to be either stable or in some cases losing activity. And so when I look at the tables and the actual data of the patients, a number of different dose groups, and at least close to half of the high dose participants lost factor VIII activity between the end of the first year and the subsequent years that they were studied. And so we don't see consistent durability in this study either. We do see good efficacy, however. And so where most of the patients are in this 5 to 40%, 30% range, we see a large decrease in the number of bleeding events from very high prior to going on to the factor VIII gene therapy to really very low numbers of annualized bleeding events following the gene therapy. So good evidence that the transgene works in this case too. Now, here's yet another study, a 4th study, looking at garaktakojin vitelaparvovec. And this is from a phase 1/2 clinical trial that has been followed out for at least 2 years in the data that are reported. In the high dose group, 4 of 5 had increases in liver enzymes. And what you can see here is a decrease from the week 52, 62%, about 50% drop at the end of 2 years. And so once again, we see that drop in the factor VIII levels that looks like it's fairly consistent between various trials. So it may be something intrinsic about the factor VIII gene that cells don't like to make it. And that's something that needs to be further investigated. I'm going to show you now some results from factor IX and concentrate really on the phase 3 study, 54 patients, that is the licensure study for AAV factor IX. And I'll show you contrasting results. So these haven't been presented in a peer-reviewed publication yet, but they were most recently updated and presented at the EHAD meeting last February from Europe. And so what we can see here is some real differences from factor VIII. First of all, this is looking at the factor IX activity over time. And what one can see through 18 months is a horizontal line, essentially. No evidence of any durability drop-off. You can look at the bottoms of these interquartile ranges and see they're all about the same. You can look at the tops of the interquartile ranges, and with the exception of this last one here, they're all about the same. And so we seem to have a consistent amount of factor IX being made, about 39 units per deciliter, 39%. And it looks as if it is durable. We also see a large decrease in the annualized bleeding rate, demonstrating superiority over prophylaxis. So these are patients that were on prophylaxis and it further decreased their bleeding rate. And so most of the patients, except for 2, were able to stop their prophylactic factor IX infusions. None returned to prophylaxis. 2 didn't respond. 1 did have a very high neutralizing antibody to the vector that was being used. And another one had an infusion reaction and only received a partial dose of the vector. There were a number of adverse events, but a small number of patients, relatively, 9, had transaminase, liver enzyme increases. They were treated with steroids, and as opposed to being treated for a median of 8 months, they were treated for about 2.5 months. And so quite a few differences with regard to factor IX versus the 4 results that I've shown you with factor VIII. Now, what else is going on here? Because it's clear that it would be very, very nice to have an alternative to AAV. The problem is we don't have an alternative to AAV. We've had a number of aborted attempts to try to come up with something else to deliver the factor VIII, factor IX transgenes, but nothing really has compared to what we can do with AAV, which is why it continues to be used in so many different trials. I want to point out this non-viral gene therapy from Generation Bio, a Cambridge, Massachusetts company. And so here's some data that they showed from a mouse, looking at the percent of normal factor VIII. And when they administer their non-viral therapy, this is their DNA, this is the DNA for factor VIII, and they stick it inside of something called a lipid nanoparticle, which can home into the liver and deliver the gene directly into the hepatocyte, just like AAV does. And so what they found with the mice is that they could boost the factor VIII level to 200% of normal, double the normal factor VIII. Remarkable result. When they tried to repeat the same thing in primates, they got to 2%. So they've gone back to the drawing board. I haven't heard much from them in the last year or so. And so this was disappointing. We'll have to wait and see if they're able to improve upon this. because in order for us to go into clinical trials in humans, it has to work in non-human primates. And this is unfortunately not evidence of working. Here's another approach from a new company here in San Diego, which is where I'm at right now, called Poseida Therapeutics. And they have also used lipid nanoparticles to deliver some enzymes that can insert the factor VIII directly into the chromosomes. And so this is called gene editing, which I'm sure you've heard about through the popular press and thousands of scientific papers by now. Nobel Prize was awarded last year for this. And so it's remarkable technology that allows us to actually insert genes directly into the chromosome. When you do that, they're in the chromosome. And if that cell divides, they go with the cells, the two daughter cells. And so it's permanent, no question about that. So this has been done in mice. And all I'll say, based upon what I just showed you on a previous slide, it's easy to cure mice. So we're waiting additional work from them to demonstrate that they can take the kinds of results that they've got in these juvenile hemophilia A mice, 50% levels of factor, and see if they can do that in non-human primates as well. And then that would be a non-viral approach, and it would allow for integration. So we would not have to worry about durability in that case. And then finally, I'll show you some results that are coming from two new companies to this field, Intellia, a gene editing company also in Cambridge, and Regeneron, a well-established biotechnology company. The two of them are working together. And what they're doing is using these lipid nanoparticles. So these are just bags of lipids that can contain the gene. In this case, the factor IX gene, ultimately, but the way they're doing this is that they're taking enzymes into these lipid nanoparticles and administering those that can cut DNA in the chromosome and allow an AAV vector to deliver the factor IX gene and insert it directly into the chromosome. And so they're also doing gene editing, but they're using in part an AAV virus to get their gene, the factor IX gene, into it. They're putting it into the albumin gene. Albumin is a circulating protein, very high levels in human beings, some of the highest of any protein in humans, and is made by the liver, by the hepatocytes. And so you could take some of those albumin genes and destroy them by putting in a factor IX gene and allowing the albumin promoter, the instructions from the albumin gene, instead of making albumin, to make factor IX. And so that's the approach they're taking. And they're continuing to progress in mice, non-human primates, and have plans to go into the clinic. So you can see that there are a number of alternatives to deal with some of the obstacles that we have with AAV. The last one I'll mention is to use a different vector. And in this case, it turns out that HIV can be a great vector to use to get permanent integration into chromosomes. One can take out the HIV genes that cause AIDS, HIV, and put in the transgene of interest, factor VIII or factor IX, and then deliver that and direct it specifically into the liver. And so that's been done by a group that's collaborating, led by Luigi Naldini, who is regarded as the father of lentiviruses since he discovered them, their use 30 some years ago now. And so there is a program that has produced some results in dogs and non-human primates, but it has not progressed into the clinic. And there are some safety issues associated with it. More importantly, it's been very difficult to make this in the quantities required for clinical use. And so we'll have to see if there are any breakthroughs on this end, but this also would provide for permanent correction of the factor VIII, factor IX gene. But there are a number of hurdles before this will become a reality. So I'm back to benefit risk and how do we think about this? And we've covered this before, but I want to return to it because it's really important to think about everything we do know, everything we know we don't know, and then to at least imagine that there will be things that occur, what we don't know that we don't know. And it makes it difficult to develop a quantitative benefit risk ratio. But we need to do the best we can. It needs to be shared decision-making, so between the patient, the patient's family, the healthcare providers, to provide as much information as needed to develop this truly as a personalized medicine. And so it's important that we look at this new technology of gene therapy in this context. We have been, a number of us have been, who either have hemophilia or used to have hemophilia, have been very active in really asking people to consider the benefit risk. But we're asking not the patients and the physicians as much as we're asking the companies who are developing these drugs. There seems to be something called Panglossian thinking. The first author on this, Radek Kaczmarek, who's a young scientist with hemophilia, came up with the term. We all had to look it up. It means overly optimistic, and it certainly applies to all of the work done with the development of AAV therapeutics, not only in hemophilia but across the board. We're all overly optimistic, and we're not necessarily looking at the benefit risk, the efficacy versus safety with the objectivity that we need to. And in this paper that we published in the leading gene therapy journal, we recommended a number of things for level setting, for advocacy, and it includes really asking the companies that are developing these drugs to spend more time on some basic research to understand how the drugs work, to figure out how to maximize the therapeutic window instead of having no therapeutic window, some efficacy when we have toxicity. Why can't we separate those out? Can we? Increasing the tropism to the target tissues and decreasing off-target effects. Moving toward gene editing. AAV therapy won't work in very young children. There's really no question about that based upon primates and mouse studies in juvenile and infant primates and mice. And that's because the liver is rapidly growing and the gene, which is not in the chromosomes, gets kicked out of the cells. And so you lose your durability. And so what are we going to do about children who would benefit the most from gene therapy? We've got a number of immunity issues that I don't have time to go into, but for most of the AAVs that are being used, they're all cousins of one another. And about half the people have pre-existing immunity, meaning we have antibodies to those AAVs that would prevent us from taking the gene therapy. So dealing with that and coming up with solutions is important. And then afterward, we can't give a second dose if we don't get a good response. It'd be great to give a second dose. Can't do that because we make a whopping immune response to the AAV. When you're injecting quadrillions of vector genomes, vector particles into the body, you're going to make a great immune response. It's a terrific vaccination. And that's not what we want to do with AAV, but that is what's happening. And then continuing non-AAV research and development along the lines that I talked about. And I want to point out how important this is. So, we think of ourselves as desiring to have gene therapy, but for 70% of the world who don't have access to most of the therapies that we are all taking here, that's a problem. And so dealing with that is important because that benefit risk changes quite a bit. So there is compensation for the lack of knowledge as these initial products go toward approval. At the WFH, the World Federation of Hemophilia, we have a few years ago developed a gene therapy registry that is about to be implemented. It's a It's an approach that has been taken with a number of both patient organizations and treater organizations in Europe and in North America. And so this is a way of tracking what's happening, an early warning system to be able to identify any adverse events once these products are out on the market, to surveil for these unpredicted events that we can't even imagine today, and to surveil for the predicted events. So the questions that need answers, I think I've gone through this, but these are the questions that a patient needs to ask in consultation with their healthcare providers, and get some answers before making the decision about gene therapy. And so I want to come back to something that a former NHF board member wrote, Steve Faust. We desire to be cured. We don't need to be cured. And so this was one person's opinion. Others may not agree with that. But what's of interest to me is that this was an opinion that was given in the leading gene therapy journal, Molecular Therapy, back in 2001. This is before we had extended half-life products, before we had bispecific antibodies such as emicizumab, and before we had any evidence that gene therapy was actually working. So it's useful for perspective because we're very fortunate here in North America. We have state-of-the-art therapies and a quality of life that is unparalleled in most other parts of the world. So what's the goal for research on hemophilia? What are we all driving toward? Well, we're like everyone else who's got a severe genetic disease, we want a cure. And the urgency for that depends upon where one lives. So we're fortunate to be part of the 15%. But for the 85%, any sort of treatment, including gene therapy, no matter how imperfect, is a significant advance. And that's important to consider. The other consideration is that for both technical and regulatory reasons, von Willebrand disease and rare bleeding disorders are being left behind at this point. And so there are people attempting to address these disparities, but there is not anything on the horizon yet. And part of it is the cumbersome nature of needing to initiate clinical trials in diseases that are ultra rare. Very hard to do at this point. We need to change the paradigm with the FDA. So coming back to the rest of the world, at the WFH, we've identified about 200,000 patients with hemophilia in the world, but there should be over 1 million. And so what's happened to the rest of those patients? Well, many of them are dead, and some others, even though they have severe hemophilia, haven't been identified, amazingly enough. So when we talk about benefit risk, what's right for a high-income country may be a different decision for a low-income country. This is looking at different regions of the world, and it's looking at the observed number of patients versus the number that there should be there. And so what we can see is in Southeast Asia, observed number should be around 30,000, is around 30,000. The expected number, though, is 200,000. Where are these patients? Well, as I said, most of them died without a diagnosis of hemophilia. So in the last couple of minutes, I'm just going to touch briefly on the rest of the therapeutic space. And it's quite crowded. We have many, many different conventional factor VIIIs. What I can say about all of these is that they all basically do the same thing, despite the marketing hype that you might hear about one being better than another. All of them are effective at stopping bleeding episodes or preventing bleeding episodes. All of them can be used 2 or 3 times a week. The doses may differ a little bit based upon the clinical trial results that have been obtained, but they're all good therapies that are safe and efficacious. On the extended half-life side, we have factors, several that have been created with different technologies. And they all have about a 50% increase in half-life, meaning that they can pretty well be taken twice a week and provide good coverage for patients. And some can be stretched out maybe a little bit longer. And then we have one new one on the horizon that's in phase 3 testing, BIV-001, which takes advantage of the FC fusion technology and adds some technology to make it independent of von Willebrand factor, which is its carrier protein. And this product has a 40-hour half-life, and so easily achieves once weekly dosing thresholds with high trough doses, high trough levels at the end of the week. So as I said, it's in phase 3. We await to see both efficacy as well as safety results from a large phase 3 study, but it offers the next generation of therapies to escape this 1.5-fold increase in half-life we see with all of these other products. On the factor 9 side, we've got 3 products using different technologies to extend half-life. All 3 of these products have been tested. They're all approved, and they all work. What I would point out is that there is some controversy that's appeared over the past year or two with the albumin fusion protein, where it appears that we're learning more and more about where factor 9 functions. And factor 9, although we measure it in the circulation, functions in the wall of the blood vessel. And so what we really need to do is measure how much factor 9 is in the wall of the blood vessel. And what has been found is some breakthrough bleeding in some patients who are receiving the albumin fusion protein. The mechanism of action for that is fascinating, more than we can cover right now, but it is something that you should discuss with your physician. And then I'll just finish up the survey of the products by looking at a whole series of drugs that are rebalancing agents. I talked about emicizumab. Emicizumab is a bispecific antibody. It's essentially a factor 8 mimetic, so it's not a rebalancing agent. But that basically functions as factor 8 functions, and as you know, that's on the market and has been shown to be very effective at providing around 15% factor 8 equivalency levels. There's more work ongoing in this area too, with further clinical testing of improved versions of this bispecific antibody. On the rebalancing side, how can we change the balance of the yin-yang in coagulation? For coagulation, we need to get proactive procoagulation, and we also need to be able to stop it and shut it down, because we don't want to be coagulating and causing thrombi all over the body. And so there's different technologies that are being used. A drug called fitusiran that targets a potent anticoagulant. So if we shut down the anticoagulants that are naturally present in the body of a person with hemophilia, we'll tip the balance and we'll have more procoagulation. That's the idea behind this and the anti-tissue factor pathway inhibitor antibodies that are being tested clinically. So these are all still in clinical trials. There have been adverse events reported with some of them that do include thromboses. And so the therapeutic window remains to be fully established. The full risk of thrombosis at the optimal dose remains to be fully established. And caution has been warranted within the clinical studies when breakthrough bleeds do occur. So the verdict is out on these. They're still in clinical testing, but they represent new alternatives and new technologies for the treatment of hemophilia, including rare bleeding disorders, I should mention. If you want more information about the state of the art for treating hemophilia, at the World Federation of Hemophilia, we have published the 3rd edition of the treatment guidelines in 2020, a series of chapters. The one I'd refer you to on the hemostatic agents was authored, well, I shouldn't say authored, coordinated by Steve Pipe. He had a team of 10 authors working on it. And so that would go into a lot more detail on the existing hemostatic agents. And this is freely available on the WFH website. There's videos of each of these authors giving synopses of the chapters as well that are present. So I'd refer you to that for a wealth of information.
Excellent. Thank you so much, Dr. Pierce.